Okay, welcome. Today we shall continue on transcription in prokaryotic, how transcription is occur in prokaryotic cell. You know, prokaryotes are like bacteria, single cell organisms. This process of transcription takes place in these organisms. So we'll see what are the mechanisms. You know, there is elongation, there is initiation actually, there is elongation and termination. What are enzymes in this process? How it happens? You know, in prokaryotic, there is no defined nucleus. There is no membrane-bounded organelles like endoplasmic reticulum, Golgi apparatus, and others. So how this process of transcription is happening in these kind of organisms? Let us continue. You know, in prokaryotic, both transcription and translations are coupled. They all occur together. They all occur together. Both transcription and translation. You know what happened? Here, there is replication. There is a replication. After that, after DNA is replicated, there is transcription. Transcription. Here, the process of transcription and both translation are coupled. They occur together. Once transcription happens, translation continues. There is no gap. There is no gap. Because there is no defined nucleus. There is no membrane-bounded organelles. Rather, here there is nucleoid. So all things happen together. All things happen together. You know, we said the information content in DNA converted into that of RNA. That process is what we call a transcription. It is a transcription. You know, there is a nitrogenous base sequences on DNA. That nitrogenous base sequence changed into that of RNA language. That process is what we call transcription. There is an enzyme we call RNA polymerase that catalyzes the formation of transcription. Then RNA will be formed. Then translation continue. The protein synthesis will occur. Look at here. This is a cytoplasm. You know, in case of prokaryotic, both transcription and translation occur in cytoplasm. Because there is no membrane-bounded nucleus. Once RNA is formed from DNA, then synthesis of protein begin. Then polypeptide will be formed on the surface of ribosome. Mind here, the information contained in DNA transcribed into that of RNA. In protein synthesis, you know, messenger RNA is very important. There are so many RNAs. Here, messenger RNA is very important. Messenger RNA carries coded information. So that once messenger RNA is formed, that messenger RNA moves to the ribosome. Where protein synthesis occur. That is all. So here, in prokaryotic, there are steps such as initiation, elongation, and termination. Let us see initiation. What happened here in initiation? What happened? Good example is E. coli. Good example of prokaryotic is E. coli. What happened here in initiation? In initiation, during initiation, RNA polymerase recognize and bind directly to the special region. That special region is, we call it a promoter. A promoter. Promoter is DNA sequence region at which RNA polymerase bind. Where transcription begins. So that is promoter. The question is, how does RNA polymerase find a promoters and on DNA? That's the question. How does RNA polymerase find a promoter? Where it is? That's the question. You know, prokaryotic RNA polymerase consists of 5 or 6 subunits. Some of them are catalytic, others are regulatory. The catalyte alpha 1, alpha 2, 1 beta prime, and omega. They are actual polymerase function. The other is sigma factor. With the help of this, RNA polymerase identify that special region. Mind you, RNA polymerase consists of 6 subunits actually. 2 alpha, beta and beta prime, omega, and a sigma factor. Let us see. So DNA-dependent RNA polymerase consists of core enzyme and a sigma factor. That is what you call hollow enzyme. Hollow enzyme is nothing but it is core enzyme plus sigma factor. Core enzymes are alpha 1, alpha 2, beta prime, and omega. They are core enzyme. They perform actual polymerase function. They catalyze the formation of RNA. Beta, it is largest subunit. The second largest subunit, beta prime, it's the largest subunit. Omega, it's very important for assembly of RNA polymerase. Alpha, third largest subunit. You have 2 domains, N-terminal and C-terminal domain. This C-terminal domain is very important to attach at the region of AT on the promoter. It's very important. So these are core enzymes. They are core enzymes. You know, RNA polymerase cannot start transcription in its own, even though there is a core enzyme. Core enzyme alone cannot start what you call a transcription if there is no sigma factor. So sigma factor is very important. It enables specific bind of RNA polymerase to promoter. To initiate transcription, that RNA polymerase must associate with a sigma factor. So sigma factor is very important to recognize that special region, we call it promoter. It brings that core enzymes, RNA polymerase to the promoter. For example, there are so many sigma factors. Sigma 17, sigma 54, sigma 28, sigma 19, so many sigma factors. Here, for example, sigma 70, it is associated with RNA polymerase. So, complete RNA polymerase, you have 6 subunits. So, it's called hollow enzyme. 6 subunits. Beta prime, alpha 1, alpha 2, omega, then sigma factor. So it is consists of these 6 subunits. Due to that, transcription begins. So during initiation, what happened? This sigma factor and the core enzyme, they form what we call a closed complex. There is 3 things very important in initiation, during the initiation process. They form closed complex. It means that RNA polymerase binds with a promoter. You know, RNA polymerase consists of 6 subunits: beta, beta prime, alpha 1, alpha 2, omega, and sigma factor. So during initiation, first they form a closed complex. After that, they form what we call open complex. Double strand start unwinding. Here RNA polymerase start adding ribonucleotide triphosphate on template DNA. That is what happened during open complex. Then finally, abortive initiation. Here, short RNA strand released. The abortive occur because sigma factor block RNA exit channel of RNA polymerase. So that sigma factor also released when nascent RNA is released, which is not mature RNA. So once sigma factor block that exit channel, nascent RNA will be released. Then after, sigma factor also released. After this, elongation begins. Once sigma factor released, RNA polymerase, particularly core enzymes, proceed what you call elongation. This indicates closed complex here. RNA polymerase bind at the promoter, then it forms a closed complex. After that, they form open complex, then abortive initiation. Then finally elongation begin. Look at here, this region. This is -35 sequence region. This is -10 sequence region. This is +1 region. Mind you, this is promoter. It is a promoter. So promoter region is consist of -35, -10, and a +1. So that RNA polymerase bind at this region. Core enzyme plus sigma factor bind at this region. They form a closed complex. Mind you, this is upstream. This is downstream. Look at here. -35 sequences, it is a recognition and binding site for RNA polymerase. You have a sequence TT, G, ACA sequence. This is a binding recognition and binding site for RNA polymerase. -10 region, it is a place where stable complex of DNA and RNA polymerase formed. -10. +1 is starting site for transcription. -10 is Tata box here. It is the place where stable complex is formed. Then at the +1, transcription begin. After that, RNA will be formed. Anyway. Okay. Then finally, it terminates the process. How can it terminate? Once RNA is formed and released, then it terminates the work. This actually depends upon specific DNA sequence. We call it a terminator. There is a terminator. This terminator triggers dissociation of transcription complex, RNA polymerase, and nascent RNA release. Actually, in bacteria, terminators come in two ways, two types: row dependent and row independent. Let us see. When I say row independent, row is nothing. It is a kind of ring-shaped protein with 6 identical subunits, hexameric. It is ring-shaped protein with 6 identical subunits. So here, termination requires the action of this protein. This protein binds to specific sequence single-stranded RNA we call RUT, raw utilization site. It binds at that site, this protein. It binds to that specific site, RUT or RUT, raw utilization site. Then the complex also binds ATP and moves along RNA, ultimately disrupting the interaction between RNA polymerase and RNA. Mind you here, once it binds here, this row either push polymerase forward relative to DNA and RNA, resulting in termination, or it induces some conformational change in polymerase causing enzyme to terminate, or it pull out RNA. It pull out RNA. So that in such a case we call it row dependent. Row dependent. What happened in row independent? In row independent, there is short inverted repeat of nucleotide, which is followed by stretch of 8 AT base pairs. They become complementary in itself. They pair with itself. So here there is inverted repeat of nucleotide, which is followed by a stretch of 8 AT base pairs. When polymerase transcribes an inverted repeat sequence, the resulting RNA can form stem loop structure or hairpin. So this rich hairpin followed by U. Mind you, A per U base pairs in DNA or RNA hybrid are weaker than other base pairs, so more easily disrupted as consequence of stem loop formation. The formation of hairpin causes termination by disrupting elongation complex. Look at here. It forms inverted repeat. Look at this strand. 8 AT. This is 8 AT. 1, 2, 3, 4, 5, 6, 7, 8. 8 AT. 8 AT. This region and this region is complementary. This is symmetry. This region and this region is a complementary. Look at here. G, C, C, G, C, G, C, G, G, C. This is at the int U. Okay? Around this area. This is CC. This is CC, UAA. So it forms a complementarity. It pairs with itself. So it forms hairpin structure. It block the elongation. So transcription folded to form termination hairpin. It folds upon itself. That is what you call row independent. The strand itself folded upon itself. In such a case, we call it is row independent. Thank you for watching this video. Subscribe, comment and share this video. Thank you very much.